So good morning, everyone, and thank you for joining us for part 2 of Azenta Life Sciences Advancing Transcriptomics, Gene Expression Screening, Single-Cell RNA-Seq, and Beyond webinar series. Once again, my name is Brianna King, and I will be moderating today's event alongside my colleague, Sibi Sabu. If you joined us for part 1, you may already know our presenter, but to those just joining us for part 2, today's webinar will be led by Dr. Andrea O'Hara, who is a strategic technical specialist within Azenta's next generation sequencing team and has more than 11 years of experience with NGS. She earned her Ph.D. from the University of North Carolina at Chapel Hill in genetics and molecular biology. Just as a reminder, please feel free to send any questions you have through the Q&A chat box located on the right-hand side of your screen. Dr. O'Hara and our Q&A panelists will address your questions during the Q&A portion of the webinar. So without further ado, I'll hand everything over to Dr. O'Hara to begin the presentation.
Thanks, Bree, for that great introduction. So today we're going to be going over part 2 of our transcriptomics series. Just a brief recap of some of the background here. So we're Azenta Life Sciences. You might previously know us from one of the many different names that we've had. So formerly GeneWiz. Back in November, we grouped together and rebranded to be all under one roof. What this means is that we offer a number of different products and services across technology here. So we have our genomics and analytical services, sample sourcing, storage, automation, and logistics, consumables and instruments, and data and informatics. And today we're going to focus again on that genomics and analytical services. All of these come with complimentary consultative services. Our genomics services are headquartered in New Jersey in the United States, in Leipzig, Germany, and in Suzhou, China. And we'll be focusing again all on the genomics and analytics side. Now, we're going to be talking today about our downstream analysis from transcriptomics. When we talk about transcriptomics, we have a number of technologies at our disposal. This is going to include Illumina short read RNA sequencing, PacBio long read ISO sequencing, 10x genomics for single cell sequencing, and nanostring genomics digital spatial profiling for that 3D resolution. If you have questions about how any of these work or how we might utilize these tools, I recommend going back and viewing part 1. I'm going to very briefly give us a recap of each of these as we switch gears between topics. But again, if you want to have that deeper dive or get more information, please go back and view part 1. So we're going to start out with our bulk RNA sequencing methods. Now, you might remember this from last time, but we're going to continue to revisit our fruit salad. So if we have a complex tissue or a fruit salad, we're going to imagine that we've got a lot of different cell types or fruits that are present within that fruit salad. And when we do our bulk RNA sequencing, we're going to take that fruit salad or that tissue, we're going to homogenize it, and we're going to get one overall flavor profile. It might be heavy in banana, it might be heavy in strawberry. It might be a strawberry banana mix. We'll be able to detect those flavor notes or those different cell types, but we're not going to be able to taste or see those very rare types. So if I only had 3 pieces of cantaloupe in there, I might not know it was ever in that particular fruit smoothie. To do this, we're going to be using our Illumina-based technologies for RNA sequencing, typically poly-A selection, but we may also use rRNA depletion or look at small RNAs. Now, when we're looking at this, we basically have our different workflows here, our high throughput gene expression screening, which is going to be for a high volume of samples, our poly-A selection, and then our total RNA sequencing, which is with that rRNA depletion. We're going to look at the analyses of the middle 2 here, which are boxed out in purple, that poly-A selection or total RNA sequencing. So let's cut straight to the analysis. Now, the RNA-Seq analysis pipeline is as follows. We're going to start looking at our sequence quality. You're going to notice a lot of these themes repeating as we go through this presentation. So once we've evaluated the sequence quality, we're going to trim and map the reads to our reference genome. We're going to generate our hit counts for our genes or our exons. The exons are going to be for the transcript level analysis. And then we'll look at our exon and gene level hit counts to do our downstream analysis. If you have questions about the very specific types of algorithms used for this entire portfolio here for the analysis, I highly recommend that you view our previous bioinformatics workshop regarding RNA sequencing, where we go into this in great detail. This is going to include all the scripting information that you might need, as well as some excellent question and answer to really review the entire analysis platform. Now let's take a look at each of these steps here so that we can actually see what we're going to get. So the first thing that we're going to start out with is our sequencing stats. This is really just going to tell us, how did our sequencing do? If we have a good high quality sample, we should have good high quality results. If we have samples that have a reduced quality for one reason or another, we may see some reduction in these statistics. And again, that really is just reflecting the sample quality. So we're going to see here in this report, we'll have the name of our sample, we'll have the barcode that was applied to it, number of reads, as well as the yield and number of megabases. And that's going to tell us how many reads we have per sample. This should be fairly normal. We'll see some variability from sample to sample, but should be kind of hitting the same ballpark target. Then we'll have our mean quality score and the percentage of bases that are over a Q30 score. These stats, the number of reads and then the quality of the reads, are going to be used as our benchmarks to make sure that everything is ready to proceed. If we have a high quality sample, we should always soar over these metrics. And that's going to be part of our Azenta quality guarantee. So if you're meeting those sample submission guidelines, you will get high quality results at the endpoint. Now, once we've looked to make sure that we have all these high quality reads, we want to make sure that they're all mapping appropriately. So the next table here that we have is our mapping statistics. And this is going to be mapping to the reference genome that you've selected. When you're getting ready to do any type of these downstream analyses, you'll provide some key information to make sure that everything is being compared like with like. So we'll have, again, total number of reads, total number of mapped reads. These should be very similar. We may have a slight difference here if we have some reads that are maybe unmapped. These can be things that are gathered from other types of sequences that were present within the sample. Once we have our total percentage of mapped reads, we'll then look at the uniquely mapped reads and we'll get some stats here. Again, this is all confirming that what we're sequencing is what it's supposed to be and that the reference that we've picked is applicable to our samples. And then from that mapping, we'll actually get our gene counts. Again, we can have that exon level or we can have the gene level. And today we're going to be focusing on the gene level analyses. Once I've generated those gene level analyses, I can start to do differential gene expression where I'm going to compare between samples and between groups. And this is where all of that big downstream analysis is going to happen. So let's take a look at what the next step is. With our RNA sequencing analysis, the first thing that we're going to do here now is actually look at our comparisons to make sure that we've designed it correctly and that our samples are normalized correctly. So on the left-hand side here, you're going to see my experimental design and sample grouping. This is going to really show us which samples I've selected and what are my groups. So I have control 1, 2, 3 and treatment A 1, 2, and 3. And I'm going to group these as control versus treatment A. So I have my individual replicates and then my two groups. The next part is normalizing the samples. We know that we did have some slight variability in the number of reads per sample. And so we want to make sure that these are all normalized so that we're comparing things that are going to be similar to one another. Here I'm showing you some box plots that are going to show the different replicates here, and they're color-coded based on their grouping. If I hover over the box plot, I'll actually be able to see all of the different values within the range here, as well as within that middle count. Next, I'm going to look at how these actually compare with regards to their profiles. This is really important because we want to make sure that our replicates are replicates and that we don't need to exclude any outliers. For standard bulk RNA sequencing, we recommend at least 3 replicates, and that's really critical so that we have the ability to exclude an outlier if needed, and that will get statistical significance. You can do more than 3 replicates if you'd like. You can do 4, 5, or 6, but we recommend at least 3. Here I'm showing you 2 different types of comparisons for our replicate analysis. So on the left-hand side, we're going to look at hierarchical clustering. This is going to show the distance between the samples along those expression profiles. So you can see here we have 2 groups. They group together nicely based off of our control versus our treatments. And you can see that the replicates themselves are similar to one another, and they're also different between the 2 different groups. So we're seeing that we do have a difference in expression from our control versus our treatments. Things that we might look out for here is if I have one sample that clusters separately from the rest, that's going to be an outlier from my study that I would want to exclude. Additionally, we might find times where everything is kind of overlaid one on top of the other. meaning that we don't see a distinction between the groups. And these are times when the samples are really similar to each other. A good example here would be a control sample that was using a vehicle that did not have treatment. So we're going to have straight up no treatment and then vehicle treated. These ones might look highly similar to one another because that control is really a net neutral. And so we might see that they don't cluster independently. That could still be an expected result, and that's okay. On the right-hand side, we're looking at principal component analysis. And this is where we're really looking at the distance between samples. You'll see here that we have the dots color-coded based on our different groupings, but we also have the replicates shown. One thing to keep in mind with principal component analysis is the X and Y axis. You'll see that we're going to use this as a recurring tool in our toolkit. However, that axis can really be zoomed in very tight or zoomed out. So when you're looking at samples, it may look like we're having a high level of variability, but that X and Y axis is really critical here because they might be highly similar, and we just have a really zoomed in view. So something to keep in mind when you start comparing PCA analyses, principal component analyses across different groupings, is to keep those axes in mind, as they can really fluctuate depending upon the comparison to comparison. Now, once we've looked at that, we can actually start doing a deeper dive on the actual data that we have, and we're going to look at the actual differentially expressed genes. So again, those previous analyses were really looking at that high level view of the sample itself. Now we're looking at this deeper dive of the genes. With all of the analyses that we've seen here, we're going to have corresponding Excel spreadsheets, lots and lots of tables of data. You'll always get your raw data back as well, but one step above from that is going to be this analyzed data. So here we're going to start out with our differentially expressed genes. This is going to be a list of all of the genes, the p-value here, fold change, and we're going to actually take a look at that in some more detail. To do this, we have two main plots that we're going to use, a volcano plot and the heat map. So a heat map here is showing the top genes that are most statistically significant and have the highest differential gene expression. You can actually adjust what that level of cutoff is and what genes you want to see here. This example here is showing us the top 30. So we'll be able to see those genes and how similar or different they are, both within the samples and then across the groupings. The volcano plot is another way for us to look at the most differentially expressed genes, those with the highest differential gene expression. And we can see that fold change as it explodes out. These are two different ways to visualize that data. But again, we may want to go back to that Excel spreadsheet itself and really look at it, the data itself, in a deeper dive, especially if we're thinking about looking at things that we know. So we want to think of this as being a comprehensive analysis, but also a discovery-based tool. So we can actually start looking at these tables and pulling out the genes of interest to see how they might look by themselves. And again, when we're looking at these different kinds of plots, we can start to filter out the data and recreate these plots with very specific sets of genes of interest. Now, our other bulk-based method that we have here is high-throughput gene expression screening. Now, with this one, again, we're looking at just that 3' end of the tail. So we're getting a gene level detection, and we're going to be looking at way more samples here. So we're typically starting with at least 384 samples. When we do this analysis, we're going to, again, start out with that same upstream workflow that we're going to start out with our sequencing stats. Now, with our stats here, we're going to get the number of reads, and then we're going to see that number of genes detected right away. That's actually happening a little bit downstream, but it's included within this summary table. The number of reads here is going to be based off of the overall target of what we picked, typically 1 to 2 million, but it may be closer to 4 to 5 million. Now, once we do the mapping, we're going to get a lot more information regarding how many genes we're able to detect. Again, this should recapitulate to what we saw in the previous table here, but we'll be able to see the number of total reads, mapped reads, number of genes detected. We're also going to look at some other stats here that are going to be indicators of quality. Percentage of rRNA, which should be relatively low, percentage of mitochondrial RNA, which is going to be an indication of quality of the sample. If we have a high level of mitochondrial RNA present, that means that there was a lot of mitochondrial lysis and that those cells, that sample was of a poor quality. And then we'll have our percent globin. And again, most of the time that should be next to nothing because we aren't typically looking at samples that contain globin. Now, once we've done our sequencing stats, our mapping, and we've generated our gene counts, that's really where the standard analysis for this type of a workflow ends. We do have custom add-on analyses, but that table with our gene counts is going to be sort of the meat of our assay. Now, if we want to do additional analysis, we can do this again on our own, or Azenta can assist you with that analysis. We're going to be using the same tools that we were using previously. So we're going to be doing principal component analysis, clustering heat maps, and volcano plots. And here I'm showing you an example of the principal clustering or principal component analysis, where you can see here the replicates and then the groupings. Now, again, we're looking at a lot more samples, but we also have a lot more replicates. So you can see how the replicates are overlaid one on top of the other with these individual dots. We can also see that the different treatment groups or the different types of samples that we're looking at here are clustering together. So we have some that are clustered tightly, some that are very different from one another. Again, something to keep in mind here with that PCA is our zoom. So we will see that depending upon how tight or how loose we go here, they may be more or less similar than one another. Next up, we have IsoSeq. And again, IsoSeq is going to be looking at the full length transcript. So previously, all of those were doing short reads, and we were looking at either just the tail end or the entire transcript, but we were looking at it within the context of a short read. With IsoSeq, we're going to be using the PacBio technology, which is going to give us a complete end-to-end. Now, with PacBio, we really want to think of this analysis as being slightly different from the rest because this is much more of a discovery-based tool. So we want to identify the isoforms that are present. We want to look at any alternatively spliced products. And we also want to see if there's anything novel that we haven't seen before. The first step will again be QC, just like previously. However, this QC is going to look a lot different from the other QCs that we looked at because we're using a different technology. So with PacBio, we're going to be looking at our CCS reads. These are the reads that we're looking at to make sure that we have the circular consensus sequence, which means that we've wrapped around that transcript and read it fully. Again, for more information about this workflow and how it's working and how it's utilized, I invite you to view part 1 of this series. So when we look at our first table here, we're going to get a bunch of summary statistics here about this actual readout, the number of reads we had, the length of the reads, and some quality scores. We're also going to have a number of different graphs and charts here at the bottom that will help us to outline this information in more detail. These are all quality-based metrics. So you'll see here on the left-hand side, we're going to have the length of the CCS read. This is telling us how long the transcript is that we're looking at. Are we getting full-length transcripts? If we had degraded RNA, we would find that this will be on the shorter side. If we have nice, intact, high-quality RNA, it's going to be on the longer side. There's an upper limit to this because transcripts typically only get to be so long. We're going to look at the CCS read quality score. So how good was our quality? Again, we should see things going as much over here to the right-hand side as possible, indicating high quality reads. Number of passes, that means how many times did it read around. And then we're going to have the overall score. All of these stats here are talking about the quality of the sample and then the quality of the readout. So if I have a good high quality sample, I should have a good high quality readout. Now, once we've completed this, the sequencing stats, we're going to actually look at our downstream analysis. Now, unlike our previous analyses, this one really doesn't have as much in regards to images because this one really all of the meat of this is going to be in the table and in that data that's going to be raw data, but also analyzed data, but a number of data files. This is where we're going to see what isoforms were detected. Do we have novel isoforms? Do we have what different isoforms are being expressed? We're really going to be thinking about what do these reads look like? What did we pull out? And how did it map back to what we know? So instead of having a number of different images here, we have to go back to these data tables and really do that deep dive. Again, this is much more of an exploratory or discovery-based tool. So we're getting a whole different type of analysis with it. Now, switching gears, let's look at single cell sequencing. So going back to that fruit salad. So we took our fruit salad or our complex tissue, we homogenized it, and we looked at that fruit smoothie or that overall bulk profile. But again, when we did that, we really missed out on anything that's rare. Do I have just a few pieces of melon? Is there any kiwi fruit present? I don't know, because it might be something that's just unable to be detected with that bulk analysis. With single cell genomics, I'm going to take each of these pieces of fruit. I'm going to look at them individually, and I can start to actually pull them apart. So I'll know how many red grapes versus how many green grapes. Did I have any cantaloupe present? Do I have any kiwi present? I'll be able to see that in a lot more detail. To do this, we're going to use our 10x Genomics platform. This is for single-cell RNA sequencing analysis. And for the analysis itself, we're going to use a tool that's called Rosalind. So Azenta, powered by Rosalind, is an online platform for downstream analysis. Again, if you have questions about the upstream steps, I invite you to go back and view part 1. I'm showing you here an example analysis with Rosalind, and I'm going to take you through this step by step. So when you log into your Rosalind portal, you'll actually start out by clicking on your experiments and then going to your experimental summary here. That's going to be your first screen. At the bottom of this page, you're going to have a list of the methods done by Rosalind in great detail, including references. This will remain the same regardless of the study that you're doing, but it'll be put up here every single time as a kind reminder. This top part here is an example report. And this is going to be a high-level summary that you would provide. So it's the opportunity for you to type in what your study was, any information you want to include, reminders to yourself about what we're looking at or what we've done here. This middle section is going to outline what the actual experiment is, and we will actually incorporate this in for you. So we're going to have the technology here. Again, this is all 10x Genomics single cell. The experimental type, it's first pre-processed through Cell Ranger and then imported into Rosalind. We'll have the species and genome build. This will vary depending upon your study. Kits that was used, number of samples, any attributes you may have provided, replicates, and then your name. Now, once we've looked at our experimental summary, again, this is just setting the tone for what we're going to look at. We're going to click over to the next tab. And this is where we're going to, again, start with the quality control. You might be noticing a theme here. So we're always going to make sure that we're having these high quality reads because that's going to impact our downstream analysis. If we don't have high quality data, we can still get there, but it might just be a little bit more difficult. So with our quality control, we're going to focus first on this table at the bottom. We're going to see our sample names as we define them, and then we're going to have total number of reads, number of cells that were captured, and average read per cell. The number of cells that were captured and the number of reads per cell are going to vary based on your study design. Typically, with Azenta, we recommend anywhere from 1,000 to 10,000 cells, and typically 50,000 to 100,000 reads per cell. Depending upon how you set up your study and what you're aiming for, again, these numbers might vary. On the left-hand side here, we're going to have a number of plots that are going to actually walk us through all of the QC measures for each of these samples. So once we've confirmed that we're hitting our numbers here as we expect, we're going to take a look at all of these plots on the left-hand side. And we can click through them one by one to get more details. So the first thing we're going to look at here is a t-SNE plot. This is a clustering-based plot where we're actually going to look at all of the samples here and make sure that they look okay, but we're looking at all of the cells for all of the samples. So we're really overlaying everything one on top of the other and taking a look at that in more detail. This is showing here each of these individual dots are the individual cells. The next of these plots are going to look very similar to what we were looking previously with our upstream bulk RNA sequencing analysis. High-level view of the overall expression profile. Again, everything's been normalized based on the number of cells and the number of reads per cell. And we're seeing that overall expression profile of our replicates across the groups. Here, instead of using box plots, they're using violin plots to show the normalization, but a similar idea, a similar concept here. We're going to see how these samples are normalized, again, based off of those different number of cells that might have been collected from sample to sample and the different numbers of reads per cell. We're going to look at that principal component analysis. You can see that replicates 1 and 2 are really similar on one side. Our controls are a little bit split. Again, not a big worry, though, because we want to keep in mind that X and Y axis. So this is a great example where we're zoomed in really tight. So it looks like there's a lot of variability, but really they are, in fact, highly similar. This plot is a little bit new. This is going to be looking at the gene bias. This is a 3' bias or 5' bias that we're taking a look at. This is really going to indicate the quality of the sample. This one looks great, but you'll see that if we do have a lot of bias, you'll see a slight humping instead of this drooping effect. But this is a way that we can actually look at the samples to see if we're seeing any degradation. Now, if you have questions about any of these plots as we're going through them, what's really excellent about Rosalind is that you're going to not only get a description on the left-hand side of how these work, but there's actually going to be an icon up here on the right-hand side, not shown, where you can actually click in and get more details, information about each of these plots and how they work. So it's a very easy to use interface, but all of these images can be downloaded directly. And again, great indications for the quality of your samples. Now, presuming that everything looks great, we can start to do a deep dive in on the sample. So when we're thinking about our single cell sequencing, we want to remember the context of this study. We're first taking that fruit salad and breaking it apart into all of the different types of fruit or the different cell types. So before we even dive into the many samples we have present, we're going to dive into the single sample. This here is showing us the single cell analysis, which is the icon on the furthest right. And we're actually going to look at one sample at a time to really identify what's happening in this sample. What are the different cell types that are present? What are the groupings I want to use? How are these expression profiles varying within the sample? So in order to do that, we're first going to cluster the cells. That's going to happen automatically, but we're going to define the clusters. Defining the clusters is going to be based off of what you already know about this sample. What cell types should be expressed? What types of profiles do these cell types have? We already go in with expectations that certain cell types will have certain expression levels of different transcripts, and that's how we identify these different cell types. So we'll start to assign the groupings based off of what we already know, what we see, and what we expect. Some might be more or less known. Some of them might be totally different cell types. Some might be subtypes, but we're going to start to divide apart all of the different groups within a particular sample. Depending upon how many samples you're doing, and what we're looking at, this might be the end of our analysis. We might really just do the deep dive on the individual sample or individual samples to draw conclusions based off of these different clusters and how they vary. This is also where we get to identify those rare cell populations that we might have otherwise missed. So for example, we might not see this little red population here in the middle if we were looking at a bulk RNA sequencing profile. Again, depending upon the analysis that you have, this is where our analysis might start and might end, but we're going to be able to generate these clusters and then get the heat maps for that different expression comparison between the different groups. Depending upon how our study is set up, we do have another layer we can add on to this. So if we click on the discovery and analysis icon, that's where we can actually start to look at normalized expression, differential expression, and then do some more search-based exploration. We're going to take a look at this differential expression. So if I've set this up so that I do have groupings of samples and I want to compare across groups, these can be groups of 1, groups of 2, groups of 3, up to us. But if I want to compare sample or samples A to B, I can take a look at those in more detail. When I look at that differential gene expression analysis, you're going to see this looks so similar to what we saw previously, because now we're going to be looking at those comparisons between the groups. So we'll get our list of genes, we're going to get that hierarchical clustering, and then we're going to get those volcano plots. Again, lots of things for us to click around and look at. We will see some pathway analysis shown here on the far right. There is so much data for us to explore within this tool, within this data set. So it's really up to us to decide where our interest lies and what kinds of things we want to gather from it. Now, one of the great benefits of Rosalind here is that we're going to get these beautiful publication-ready figures. We'll be able to download those as we go through it. And also, we'll always have access to our raw data. So not shown is this icon here with the cloud, where we actually can download all of our raw data, but we can also download these images and tables as we go through the analysis. Switching gears to digital spatial profiling. So again, coming back to our fruit salad, we looked at our bulk analysis, or that fruit smoothie of the fruit salad. We looked at our single cell analysis, taking that fruit salad, sorting out the pieces and looking at them one by one to see the profile of each individual piece of fruit or each individual cell. But what about looking at it within the context of that tissue or of that fruit salad to see what types of fruits are touching what types of cells are next to one another? To do that, we're going to look at the NanoString Genomics Digital Spatial Profiler, or DSP. This is going to allow us to do spatial RNA sequencing. Again, if you have questions about the application, how we use this, the setup of the approach, I invite you to go back and view part 1. So the first part here, just like all the rest, is that we're going to start out with that QC and normalization, and then we're going to look at our downstream analyses. Now, the normalization and QC is going to be slightly different for this because of the type of analysis that we're looking at. So if we're doing the RNA sequencing, we're going to do a gene level normalization using our housekeeper genes. It's going to help us normalize the expression level. We're also going to do an area normalization. So with this, we are going to pick regions of interest, or ROIs. And we want to make sure that we're looking at normalized amounts so that if we have something that's bigger or smaller or somehow custom from one another, that we're able to compare like to like. So that means that first we're going to look at some images of our region of interest selection strategy. This is just an example. There's a lot of different selection strategies you can use, again, covered in part 1. But so for this particular example study that I'm showing you here, we're going to have 3 regions of interest. You can see they're not necessarily the same size, but over here they're zoomed in so we can see what these different cell morphologies look like and the staining patterns. Now, based off of that, we're going to do our QC, and we're going to actually look to make sure that we are normalizing the data and that everything passes QC. So you'll see here at the bottom, I have this table showing our QC status along with some stats associated with it. Once we have the data that's been normalized based off of the gene expression and the regions of interest, we can then do our downstream analysis. Hopefully you're recognizing a theme here, because our downstream analysis is going to be using the same tools that we used upstream, although now in the context of the ROIs. So we're going to start out with a clustering-based heat map. We're going to have this based off of the regions of interest and the slides, and we're going to see how they cluster map together. Now, again, depending upon how we have this set up, we could have examples where the regions of interest might cluster together, but not necessarily based off of slide, or the slides might cluster together. Really depends on how our design is set up. There are going to be times that we would anticipate that ROI 1 might be the same between slide 1, slide 2, and slide 3, and then ROI 2, ROI 3. There might be other ways that we design this study so that we would expect that all of slide 1 to look mostly similar, all of slide 2 to look mostly similar, all of slide 3 to look mostly similar. Depending upon your study design, that's really going to tease apart, when we look at this, what makes sense here. So I know we're seeing some differences down here in the bottom, but again, that might be by design based off of your study analysis. Next step here is that principal component analysis. Again, we're looking at this based off of not just slides, but also regions of interest, so we can actually see how they cluster and separate apart. Unlike the other ones where we saw this in a 2D setting, this is in a 3D setting, so there's actually going to be X, Y, and Z level axis. And then finally, we're going to look at that volcano plot to look at the highly differentially expressed genes. So we're really looking at the same analyses that we were looking at previously, but we're now doing it in the context of that 3D space. Now, last up, we have proteomics profiling. Proteomics profiling is really shifting to a whole new gear, because with all of our previous ones, we were looking at transcriptomics or RNA level expression. With proteomics, we're looking at the protein level expression. You want to think of this as the icing on the cake, the cherry on top. We're going to use this to validate what we previously saw in our upstream analyses. This is a really great way for us to show with certainty that certain proteins are present or absent that we already identified with our other upstream studies. To do this, we'll be using Olink. Olink is a great technology. It's very sensitive and has high specificity. For more questions about how Olink works, again, I invite you to go back and view part 1. Now, with our Olink analysis, we're actually going to have 2 different reports that are going to be provided. The first one is our Azenta designed report, and this is going to include study design and general QC. Again, themes that we were noticing already, we really want to make sure that everything is having a high quality result. QC is always critical in any analysis that you're doing, because all of those downstream analyses depend on having high quality upstream data. So we're always going to focus on looking at the QC first to make sure that it fits. The great thing about the Olink assay is that there are a number of different quality controls that are built into it. So in addition to looking at stats, there's also a number of internal controls. Things to keep in mind with our previous studies is that we always want to make sure we're including appropriate positive and negative controls as needed. For Olink, we're going to start out with this Azenta-based report with our plate map and study design. This is going to include our sample controls, positive and negative controls, calibration samples, empty wells, a good visualization for what we expect to see across our analysis. We have a number of quality control metrics that are provided, the observed value as well as the acceptance criteria. And this allows us to make sure that we have passing samples and that everything is what we expect. We're also going to have a review of the internal controls. As I mentioned, Olink comes with a number of internal controls that are provided with the assay. So we want to make sure that we're seeing excellent values across the board. Now, again, the Azenta-based report here is really focusing on study design and quality control. There's a secondary report that comes with Olink, which is the certificate of analysis. This is going to be a PDF file that's going to go through additional QC metrics, including some that are not shown here, as well as an attached Excel file that's going to show the normalized protein expression data and, depending upon the assay, absolute quantification. This is a way for you to actually see what is or is not being expressed. What you see here will vary depending upon which assay you select. So if we put this all together, we actually have a number of different platforms here, but we're going to see similarities with regards to what kind of analysis we can do, but also distinctions in how that works. So for bulk RNA sequencing, we're always going to start with trimming, mapping, and normalization. This is going to be true for our mRNA sequencing, as well as our high-throughput gene expression sequencing. For mRNA, we'll get transcript and gene level analysis. For high throughput, we're just looking at gene level analysis. That's where the standard analysis ends for high throughput gene expression screening. But for this standard RNA sequencing, we take this a little bit further. We're going to look at our sample replicate analysis, as well as our group-wise comparisons. And this includes PCA, which is principal component analysis, clustering, and volcano plots. For our single cell RNA sequencing, again, starting out with that trimming, mapping, and normalization, we're going to have our transcript and gene level analysis, and then we're going to do a deeper dive. We'll first start within samples to look at that cell type analysis, really looking carefully at all of these expression profiles within the individual cell. Then, depending upon our study design, we can do group-wise comparisons. This can still be done group-wise within the cell, but we can also do cross-cell or cross-sample comparisons. And with these analyses, we're going to be using the same tools, that PCA, hierarchical clustering, and volcano plots. Spatial profiling is doing this now all within the context of that tissue. We're going to start with our trimming and mapping, and then normalization, not only based off of that gene expression profile, but also based off of the regions of interest. We're going to look at transcript and gene level analysis. We'll look at our sample replicates, and then we'll do our group-wise comparisons. Again, same analyses, PCA, clustering, and volcano plots. Taking all of these results together, we can go from one to the next, depending upon the sample type, but we might pick and choose which ones we'd opt to use. Once we've completed our transcriptomics analysis, we're ready to go into our biomarker discovery and confirmation. And with this one, we're going to, again, start out with QC, different types of QC, though, because we're now looking at proteins instead of RNA. And we're going to look at our reports. Everything will still be normalized, and we can get both relative and absolute quantitation depending upon the assay that we're looking at. Now we know that you have a number of different providers to work with, so why would you want to choose Azenta? We're offering you greater efficiency with less effort. So we have discussed today really focusing again on our RNA sequencing platforms, but we do have a wide variety of different platforms that we cover and solutions for really any type of project. So if you have any questions about any of the other technologies not discussed today, please reach out and let us know. We have one of the highest on-time delivery rates for next-generation sequencing, and we have a ton of experience with all of these assays and more. If you have any questions or want to reach out further, please do let us know. And with that, I'm happy to take any questions and get our roundtable discussion started.
All right, great. Thank you so much, Dr. O'Hara, for that deep dive into the analysis of these results from transcriptomics approaches. So we did have a few questions trickle in, so we'll just start with the Q&A portion of this session, which will be led by Sibi.
Yes, thank you, Brianna. Thank you, Andrea, for that presentation. As we get ready to answer some of the Q&A questions, I think it's appropriate if we kind of introduce ourselves. I'd like our panelists to kind of take a moment to introduce themselves while we get more questions. Anthony, maybe we can start with you. Could you give us a little bit of introduction about yourself, and then Zane, Renee?
Yeah, absolutely. I'd be happy to. Hi, everyone. Thank you so much for joining us. So my name is Anthony Moore. I work here in the Next Generation Sequencing Department of Azenta. I've been with the company over 2.5 years now. I'm located in New Jersey, near where our NGS headquarters is for America. And yeah, I look forward to answering your questions.
Hi everyone, my name is Zane Alvey. I'm a senior bioinformatics scientist with Azenta. I've been with the company for a little bit over 5 years. I do lots of data analysis, troubleshooting, and onboarding of new employees. And yeah, looking forward to answering any of the questions that you guys have shared with us. And on to you, Pranay.
Hello, I'm Pranay Vishwanath. I'm a bioinformatics team lead on the Azenta team. My day-to-day responsibilities largely consist of analyses for projects, as well as maintaining and enhancing our bioinformatics workflows and pipelines.
Awesome. Well, great. Thank you, everyone. The first question, when we just dive in, and Anthony, I'm going to ask if you can help us get this started off. If customers do encounter certain challenges with their data analysis, could you speak to a little bit of, whether or not we offer bioinformatics support, excuse me, post-delivery or on a previous project? Are we able to even help them even if they haven't done that sequencing with us?
Yeah, absolutely. That's a great question, because the way we operate at Azenta is, even if you initially didn't get bioinformatics with us, let's say you're going through, you're trying to analyze the data in your lab. If you decide that you want BI support, we'll be happy always to come in and give BI support down the line. So all you have to do is reach out to the project manager that you'll have for the project, and we'll be happy to talk about what type of bioinformatic analysis you would like, and we can go back and add that to the project. And that's the same with the, let's say if you had your project sequenced somewhere else, but you want to bring it to us to do the bioinformatics, we can certainly do a standalone bioinformatics project. You'll just send us the data, talk to us about what you're looking for, and we can get you set up on performing the bioinformatics. And so, yeah, I'm very proud of our post-delivery support. And all of these initial consultations are free of charge.
Just to add on a bit to that, I should also add that we do offer a certain level of customization to whatever analyses we provide as well. So even if the analysis that you're requesting doesn't precisely meet something that we offer in-house, we're always happy to talk to you and try and determine what exactly we can perform and what exactly we can deliver to you that would best answer your analysis questions. So in that regard,
Yes.
we also have a fair amount of flexibility with what we can offer you.
Yes, I'll also like to share that if sometimes we might not be able to do the analysis in-house, or the customer will guide you guys towards the right direction using some free tools or online resources that you can probably utilize to get the answer that you're looking for as well. So we're always willing to help you guys solve your questions in need.
Awesome. And Zane, maybe you can take this. When the project is completed, what sort of BI deliverables can our customers expect? Could they request sort of reanalysis after we've done sort of analysis on their data? Yeah, could you speak to that a little bit more?
Yeah, sure. Our standard deliverables for all our projects, it includes the FASTQ files, which is the raw data files. Usually, depending on the project type, they'll be trimmed or untrimmed or raw data. In terms of being able to reanalyze your project, we're always happy to reanalyze your project. Sometimes, if you guys would like to do some more comparisons for differentiate bulk RNA-Seq analysis, we're glad to add those to reanalysis. We have done multiple times. But if you have like a whole new reference or something else, we might have to go back to a new quote or do where we can help you guys too, like with a minimal fee or on a discounted rate, depending on the situation. And aside from this, we can also combine references or do anything of that nature as well if you guys need to.
Yeah. Awesome. Another question around, since we're talking about sort of analyses and sort of deliverables, when it comes to like comparison replicate analysis, like heat map, PC analysis, are we looking at sort of the top 100 differentially expressed genes of each replicates, or other parameters for each of those replicates?
Sure, I can take that. So our standard reports and our image that we publish, we do the top 30. It's based on the for bulk RNA-seq, it's based on the wall test and the parameters we set with the adjusted p-value and the absolute value of 1. Basically, negative positive 1, basically, whatever falls outside of those. Which of those parameters will be presented in those heat maps. Of course, if you are looking for something else, we can, depending on the situation, we can always discuss and talk with our developers and do some custom stuff as well. So
Awesome. And Pranay, for triplicates, are we suggesting biological or technical replicates? I know many times we run across that issue of like very limited cells, and will they be able to perform, say, an RNA-seq experiment with just biological duplicates rather than triplets?
So where possible, we usually like to have at least a few biological replicates for each condition group that we're working with. The addition of these data points provides us with a little more statistical power with which we can call various differentially expressed genes or differentially expressed features that we can take into account. While technical replicates could be used to generally assess various other sequencing parameters, when we're looking at the actual RNA-seq analysis that we perform, we generally like biological replicates where possible. We can certainly perform without as well, but that significantly reduces the amount of power that the analysis has.
Yeah.
And the only thing that I would add to that is, obviously the downstream goal of a lot of these studies is to publish. And if you're doing an experiment in which you're relying on technical replicates or in which you don't have enough biological replicates to make strong conclusions, a lot of journals would take issue with that. And so, you're kind of setting yourself up for success by making sure that you include enough biological replicates in with the project.
Yeah, that's a great point. And Zane, can we have more than triple data sets for, say, bulk RNA-seq? Would we be able to have a single biological replicate per group for biological RNA-seq analysis? Differences in analysis? Could you speak to that?
Sure. So if you have more than more biological replicates for your per group, that'll give you more accuracy and sensitivity when trying to do your analysis, which is excellent. And if there's some samples that fall out because of sample prep or contamination or whatever it could be, like for cDNA quality, we can remove those post-analysis and then redo the analysis for those comparison. But in case of having just one biological replicate per group, in our experience, most folks who do those type of studies are usually doing as an exploratory study, and they usually have some other things that back up, like fluorescent microscopy to back up their cell data or other cell culture results that could potentially lead them to the right direction. And for publication needs, they usually just publish those data as like an exploratory study, and they'll have like a follow-up study or publication where they'll do more biological replicates and at the same time. So we have seen that. But once again, in terms of analysis, having more biological replicates is very important, and that's what we recommend. If not, it could be exploratory study to summarize it.
Yeah. And since you kind of touched on contamination, we had a question around that regarding whether or not we'll be able to remove contamination in bulk RNA-seq analysis. Are we able to add custom genes to a reference for bulk RNA-seq analysis? And whether or not we do transfected samples with bulk RNA-seq analysis. Anthony, Zane, whoever.
Sure, I'll take the first hand. So for transfected, that's either we do have a human mouse xenograft reference already established, which we have used multiple different times for multiple clients and has generated amazing results. And as for contamination or adding genes, for adding genes, it's very simple. If you have like a human sample and you want to add some genes or remove some human genes and add like a modified gene that you guys have inserted, we can do that as well. And then on top of that, for contamination, our process is basically, it's multiple different ways to do that. It's a very good question. Our basic routine is usually map to the contaminated sample first, take the unmapped reads, and then map it to the desired reference of choice, basically, and then do the analysis moving forward from there, and then that'll be the final result. And we can, if you guys are willing to, want to, we can deliver the unmapped reads as well, so you have clean reads that are going towards your desired reference as well, if you need to.
Yeah.
As well. So.
Awesome. And Pranay, regarding sort of,
would you mind if I add to, the only thing I would add to that is just always make sure to let us know when you have these issues. Like if you have a gene that you inserted in, like you just need to make sure that we're aware of that beforehand. So that way when we're setting up the bioinformatics, we can go ahead and add that in. Or if we do need to align to get rid of reads, sometimes researchers won't let us know these things till after the fact, so it can end up delaying the analysis. So we can help you the best as long as you're proactive and let us know that you have these concerns about how we can analyze the data.
Yeah, good point. And Pranay, speaking of like minimal number of reads recommended, specifically around bulk RNA sequencing analysis from one of our attendees, is there a difference between sort of eukaryotic species like human versus prokaryotic species like E. coli that we would recommend, that you can speak to a little bit?
So generally for most bulk RNA-seq experiments, we can typically get away with about 15 to 20 million reads per sample for eukaryotic organisms. Naturally, for simpler organisms like bacterial samples, we can reduce this number depending on the level of coverage we're shooting for, or depending on the size of the transcriptome of the organism. So largely, what we've seen in past projects is that this tends to kind of scale with complexity.
Awesome. Yeah. And one of the, I think, cool topics is around sort of multi-omics, and sort of what we can do to help our customers in that area. Zane, could you speak to a little bit more of like what type of multi-omic analysis we do offer, and whether or not we're able to help with overlaying any sort of like ChIP-seq and RNA-seq data and beyond?
Yes, of course. We do independent like RNA-seq analysis, bulk RNA-seq, and ChIP-seq, methyl-seq analysis as well. Aside from this, we also do genome assemblies, transcriptome assemblies, and annotation of those assemblies as well. This includes for PacBio and for traditional Illumina sequencing as well. So we have plethora of omics data. If we have multiple times we have gotten data where clients will do transcriptome assembly of their samples and then do RNA-seq analysis on top of that, and then even do variant analysis on top of that too, to see what variants are being found and what genes are being expressed or what context they have assembled are being expressed. And then they can correlate that to the annotation that we have done previously for the transcriptome assembly. But yeah,
awesome.
We do plus. Yeah. And if you have other ideas, then we're always open to do omic studies with clients at any given time. So just reach out to our study managers.
Yeah, awesome. And we have time for one more question, and I think this kind of ties into our previous webinar, part one that we kind of led into. And Anthony, I'd like to direct this to you, and we can wrap it up with this. Difference in sample preparation, specifically around bulk RNA-seq and single-cell RNA sequencing. Could you speak to that a little bit?
Yeah, absolutely. I think that's a great question because a lot of people, are familiar with bulk RNA-seq and might be exploring doing single cell at the same time. And they need to know, like, are these samples going to work for single cell? With bulk RNA-seq, you can make an RNA-seq library out of almost anything, a small frozen cell pellet, small piece of tissue. There's a lot of flexibility in terms of what you can extract total RNA from. With the single cell experiment, you're typically loading live cells into the 10x chromium. And so you need to think that, like, are these cells going to have a high enough viability that they'll be suitable for a 10x experiment? And so one thing to consider is you're not going to use a flash frozen cell pellet. You need to make sure that you're cryopreserving it down so you don't get these ice crystals forming that are going to pierce the cells. So that's very important to think about when doing a single cell. There is some flexibility there. There's also single nuclei experiments. From there, you can start with tissue and you can extract a nuclei directly from the tissue. to kind of sidestep the viability concern. But often with 10x experiments, it's going to be about having viable cells. It's very crucial to making sure that you can generate good data from the experiment.
Awesome. Cool. So with that, Brianna, I'd like to hand it back to you.
Great, thank you. And thank you to all of those who submitted questions, to all of our attendees for joining us, also to Dr. O'Hara for her presentation, and our Q&A panelists for joining us to answer some questions. If you would like to view part 1 or part 2 again, we will be sending out the recordings via email next week. But thanks again, everyone, and we hope you enjoy the rest of your day and hope to see you for some of our upcoming webinars. Bye.